Hello viewers, in this particular lecture of microbiology, I shall be discussing the different aspects of historical perspective of immunology, where I shall be discussing between the period of 1942 till 1968, and this period primarily will encompass the different aspects like the development of antigen, antibody, the interrelationship of antigen, antibody, development of vaccine, adjuvant, etc. To start with, in the year 1942, it was Freund and McDermott. They discovered a unique principle which is called adjuvant. Adjuvant molecule may be of different types. It may be organic or inorganic compounds like aluminium salts, it can be paraffin oil, or even it can be bacterial cell, dead bacterial cell. What happens is adjuvants basically increase the efficacy of the vaccine. You can see here that this particular vaccine with the adjuvant will enter, it will form an endosome and it will safely be landing into the target organ. So the activity of the vaccine increases and the actual hypersensitive reaction in the form of inflammation or other antigen processing also is initiated because of the presence of adjuvant.
Sir, I have a question. Why aluminium salt is used as adjuvants?
You see, aluminium is a light metal, it is less toxic, and at the same time it can bind well with the antigenic molecule or with the vaccine molecule, as a result of which it gives more stability. So obviously aluminium will ensure that the vaccine will be stable for a longer period and it can reach the target organ. Okay. Now in the year 1946, it was George Snell and Gorer. They discovered a unique compound which is, or unique component which is called major histocompatibility complex. This MHC complex actually is encoded by a large gene family and they basically mediate reaction with the WBC, and this tussle will ensure compatibility of a particular foreign cell into human body. So MHC complex basically is divided into three classes, and what happens is you can see that the MHC is produced inside after the processing of the proteasome, and the MHC will bind with specific chaperone which will lead to its modification, and then it will be produced in the ER and generated into the Golgi, and this Golgi vesicle will take it to the cell surface. So in the cell surface they will bind with the TCR and will bind with the specific CD8 or CD4 T cells. So MHC that way is helping in the binding of specific antibody or CD4 cells. Now basically there are three types. The MHC class 1, which is having the alpha 1, alpha 2 and alpha 3, and the beta microglobulin. MHC class 2 having also the alpha 1 and beta 1 and alpha 2 and beta 2 components, and also the MHC class 3 having the alpha 2, alpha 1, beta 2 components. But you can clearly see that these molecules are different or distinct from each other, though the common feature is that they are always binding with the cell membrane. Now actually MHC is having a specific human leukocyte antigen, or it is also called HLA for human. And it is also very important to know here that MHC will recognize specific antigen, and thereby it will determine that which particular antigen is recognized. So this particular organ will be recognized and this will not be recognized. Okay. Now we come to the next major discovery by these three gentlemen, Enders, Weller and Robbins. They actually showed that there is a particular virus which is the polio virus. The poliomyelitis virus causing the dreadful disease of polio, and vaccine there, but this virus cannot be grown in culture. So these three gentlemen grew this particular polio virus. You can see these are the polio viruses growing in the renal tissue or the testicular tissue of monkey. So culturing of this particular tissue actually denote that this particular polio virus can be of different types, and this was also done by the neutralization test.
Sir, I have another question.
Okay.
Why is neutralization test done for poliovirus in monkey?
You see, it can be done in monkey, it can be done in anybody else, but basically neutralization test will denote that which particular antibody will bind with which particular antigen, and thereby it can detect specific strain of viruses. So neutralization meaning binding of specific antibody with the virus, which will classify the polio virus into different types. Now coming to a very important point denoted by Owens in 1949. This is called the immunotolerance hypothesis. It was denoted in a dizygotic twin in cattle. And what is that? Actually, it means immunoresponsiveness loss or minimum immunoresponsiveness to foreign antigen. That means whenever the antigen is there, my antibody will not be reacting to that. In one way, I am immunocompromised. So this particular immune responsiveness can be of two types. One is actually the central, which is mediated by the thymus, which is mediated by the bone marrow. So specific antibodies or the T lymphocytes which are produced will recognize the foreign antigen. Or it can be peripheral. That is, by the peripheral lymphoidal tissue, it will generate or it will understand a particular antigen. But what is important here, this theory specifically indicate that okay, there are specific, you can see antigen binding with the antibody. But this story doesn't end here. If the responsiveness is lost, then there is a possibility that we can suffer from immune suppressed diseases, or we call it autoimmune disorders. And these diseases can include the SLE or systemic lupus erythematosus, or it can be X-linked polyendocrinopathy, or it can be other autoimmune disorders like the rheumatoid arthritis. So this is not a good prognosis, but at the same time, this story of autoimmune disorder and its explanation comes from this particular theory. Now coming to the next important discovery by Theiler in 1951. Theiler actually developed the famous or the dreadful disease vaccine called yellow fever. This yellow fever vaccine was developed from the 17D attenuated yellow fever strain, and initially when it was discovered, he actually said that the disease can be totally eradicated by repeated shot. At least 6 to 7 shots should be given of the yellow fever vaccine. But later on, WHO resulted or discovered a better shot of this vaccine, and nowadays a single shot of this vaccine is adequate enough to treat the disease. In the year 1957, Barnett made a unique discovery. This discovery is called the clonal selection theory. Actually, it is based on the discovery of Jerne earlier, who stated that there may be a large number of array of lymphocyte, but with a specific infection, a specific set of lymphocyte is active. So this led to the clonal selection, or which particular clone of this antibody will be developed to combat a particular infection. This was the basic essence of this theory. And the theory goes like this, that each lymphocyte bears a single type of receptor with a unique specificity. Receptor occupation is required for cell activation. The differential effector cells derived from activated lymphocyte will be a receptor of identical specificity, and these lymphocytes bearing receptor will be deleted at an early stage. So this is basically the theory. But when we get into this particular figure, you can see that the specific clone of B cells are produced like this. That is, B cell will bind with an antigen, it will respond to the antigen, it will proliferate a series of similar clone. You can see that a same number of this clone is produced, and ultimately they will active the plasma cell to produce specific antibodies. So basically the clonal selection theory will indicate a specific antibody against a specific antigen, and which is highly clone specific.
Sir, I have a question. What is the role of VDJ recombination?
You see, VDJ recombination will make the antibody specific for a particular antigen. So a VDJ recombination, it will undergo this recombination to make the antibody specific for an antigen. So there is a perfect binding between the antigen and the antibody. So that you can see that antigen binding site becomes specific for that particular antigen. Okay, good. Now coming to the next important discovery in 1957, it was Isaac and Lindemann. They discovered a unique molecule which is a glycoprotein, and this glycoprotein is called the interferon. And this interferon, basically they prevent a specific viral attack. How? By recognizing a particular virus, they will activate the natural killer cells, they will activate the macrophage, and thereby the defense reaction will be activated. Interferons can be of three basic types. Primarily the two types are IFN alpha and IFN beta, but there is also a third type which is the IFN gamma. And now the IFN gamma, you can see the multivarious activity of this IFN gamma. On one side it will activate the NK cell and the T cell, or at the B cell, or at the same time it will activate the macrophage, it will activate the other granulocytes. In one way, it will make the body more prepared for an infection, or rather, in other words, it will combat an infection at the earliest stage. Subsequently, it was Dausset in the year, it is a long period, basically between the year of 1958 to 1962, he discovered a particular human lymphocyte antigen locus. And this locus was discovered in the chromosome 6. You can see there are three specific locus. They call it human leukocyte antigen 1, human leukocyte antigen 2, and human leukocyte antigen 3. Basically, these particular polypeptides can be processed by proteasome, and they will be helping in the stimulation of the T cells and the B cells, and they will ultimately bind with the natural killer cell or bind with the antigen presenting cells, or will activate the helper T cells and provide immunity. So basically, this particular group of HLA class 1 and class 2 will make your body more protected against viral infection. In between the year 1959 to 1962, these two gentlemen, namely Edelman and Porter, they denoted a unique structure of antibody. So the antibody structure, the antibody function was somewhat known, but the structure was denoted by these two gentlemen. It is a typical Y-shaped structure having the variable and the constant light and heavy chains of protein, and it is also having the stem, and they are interconnected by disulfide linkage, and basically there is a specific antigen binding site, and you know this particular site is having this VDJ recombination so that the outer topology changes of that antibody molecule. And there are specific properties which are also denoted by them. They actually showed that antibodies are produced by specific WBC or white blood cells called plasma cell. Antibodies are glycoprotein, and they belong to a particular immunoglobulin superfamily, and they have the typical light and heavy chains, and they are having a specific antigen binding site. And they are, of course, they are attached to a particular membrane, and they can be attached to polymorphonucleosides. They can bind to specific tyrosine enriched receptor. But what is the importance of that? They will actually follow a particular target. So that means it is a guided molecule which will guide the particular cell to a particular site. So often antibody labeling will help in target specific release of vaccine. Coming to the next important discovery. Now this discovery was by Gowans in the year 1959. He showed that the lymphocyte circulation follow a unique pathway. Now you know lymphocytes primarily they may be the B cells produced from bursa or T cells produced from thymus. They are primarily flowing through the blood vessel, so they are moving in the circulation and they contain the T cells, and then they come outside the blood vessel. They come into the interstitial space, And when they come into this interstitial space, they can get into spleen, they can get into lymph node, they can get into mucosal sites, or even sometimes in the lung, in the macrophages. And these particular cells will sometimes be enriched. So sometimes the lymphocytes which are, say, less in number in the lymph node, the production may increase. Or sometimes the lymphocytes which are no longer in function will be destroyed in the spleen. So in other words, the reinforcement is done in this lymphoidal tissue, and after reinforcement, the lymphatic duct directly open into the bloodstream. And actually, it is the subclavian vein which receives this lymphatic. And this particular lymph cells or lymphocytes, including the B cells and the T cells, will circulate again and will get into the specific infectious site. So this particular cycle, namely the lymphocyte circulation or lymphocyte circulation type, was denoted also by Schnuder much later in 1978, who denoted that the recirculation from lymph node to peripheral blood becomes most important in case of infection. So in other words, the direct prevention is very important at the outset of infection, which was denoted by Schnuder. Now between the year 1961 and 62, it was Miller who denoted the role of thymus. Now thymus is a unique gland. It is substernal in position. It is somewhere here. This gland actually produces the T lymphocytes. This is the thymus, what it looks like. But what is important here, that this thymus gradually with age becomes smaller. And this particular T lymphocytes later on is not always produced by the thymus, but it comes from the bone marrow. But this particular thymus will produce the T cells, and the T cells will be of different types: the helper T cell, the memory T cell, the cytotoxic T cell, which ultimately will help in protection. And this particular protection actually can be peripheral or it can be central. When it is peripheral, it is produced by the peripheral lymph node, and when it is central, it is produced by the thymus and other bone marrow. Now it was Peter Nowell, actually he in the year 1961, he showed that these lymphocytes may be activated by the incorporation of certain hormone, which include the corticotropin releasing hormone, or straight away I can say it can be stimulated by certain steroid hormones like the corticosterone. Now how it does? Actually, the administration of CRH will increase the IL molecule, interleukins. It can also activate the CD4 cells. It can increase the production of IgH, or it can actually mediate the production of both IL-1 and IL-2. And basically I can say in one way the hypersensitivity or cell proliferation of lymphocyte increases with the administration of glucocorticoid. So in one way we can say certain activities of defense is increasing with the administration of corticosterone, but certain activities are also down regulated, like the production of IFN gamma, like the production of LPS mediated IL-1. So what does it mean? It means sometimes that steroid can apparently change or modify the defense reaction in our body. This is a very important discovery. Now coming to the next important discovery, which was by Kamakura and others, who in the year 1965 denoted that lymphocyte may actually be produced from the thymus, the T cells, and they mediate cellular immunity or cell mediated immunity, and there is always the B lymphocyte which will mediate the humoral immunity. But these two types of reaction actually is definitely mediated by the different types of T cells. And not only that, this particular lymphocyte can assist in the destruction of certain cells at the time of immunity, or it, in other words, it may act as a blastogenic factor.
Sir, I have a question.
Yeah.
In what way the blastogenic factor imparts protection?
You see, blastogenic factor means the cell will be blasted, the cell will be destroyed, destructed. So if there is a cell which is containing infectious viruses, so if it is destroyed, the viruses will be released and the viruses will be targeted upon by the other antibodies. At the same time, if there is a tumor inducing cell or the tumorigenic cell, that also can be blasted, destroyed. So that way it is imparting protection. Clear? Now the next important discovery was by Wheelock and others in the year 1965, who denoted the structure of immunoglobulin G or IgG, and this is the structure of IgG. And you know, this molecule is very important because it is actually responsible for defense against any foreign bacteria or virus. It is the major antibody which is produced against any infection. Now this gentleman, that is Ishizaka, in the year 1967 denoted the structure of another antibody which is called the IgE, immunoglobulin epsilon. And this immunoglobulin epsilon actually is called the allergic antibody. So if there is any particular allergic reaction, the mediator is this IgE. And coming to the last major discovery of this particular lecture by Elkins and Gutman in the year 1968, they wanted to explain the mechanism of allorecognition and rejection. What is that? Whenever there is a particular tissue grafted, in what way it will be recognized and rejected. It can be either direct, where the antigen presenting cell carried in the donor allograft will migrate to the recipient lymphoid. So it is called direct because it is direct migration. And in sometimes it can be indirect, meaning there will be the precursor antigen presenting cells which will be produced from the bone marrow, and they will travel to the allograft, and there will be signals which will take place in the allograft, and thereby rejection will occur. But the question is, it takes time. So direct allorecognition is faster, but indirect allorecognition is, it mediates organ transfer or organ rejection at later time, but it takes time. So in one way we can say that this allorecognition and its rejection is very important for organ transplant reaction.